For this experiment, you will need a strawberry, a Ziploc bag, a strainer, tweezers, measuring cups, measuring spoons, a beaker, 90 milliliters of water, 2 teaspoons of dish soap, 1/4 teaspoon of salt, and chilled isopropyl alcohol. We chose to use strawberries in this experiment because strawberries are octoploid, meaning they have 8 sets of chromosomes in their DNA, whereas humans only have 2. This provides the strawberry with a lot of DNA that will end up sticking together in the experiment, ready for us to extract. The fact that strawberries are octoploid also increases the possibility for the success of this experiment, since there is a lot of DNA that exists for us to extract. So why is dish soap necessary for this experiment? Well, dish soap is actually amphipathic, which means that it contains polar and nonpolar components in its structure. This characteristic allows it to bind to the lipids of the cell membranes and actually separate the phospholipids in the cell membrane of the strawberry. And this causes the cell membranes to dissolve and the DNA to escape, so we can extract it.
The DNA sugar phosphate backbone is also polar. Contrarily, the isopropyl alcohol is nonpolar, which means that the DNA molecules are not soluble in the isopropyl alcohol. Therefore, this further causes the precipitation of the DNA, meaning that the DNA clumps together and then forms a solid that can be seen, that becomes visible in the solution, since the isopropyl alcohol is not attracted to the DNA itself. So you may be wondering what the salt does in the experiment. So basically, the DNA backbone has phosphates, which have a negative charge. So when you add salt, which dissociates into its sodium and calcium ions, the sodium ions have a positive charge that neutralizes the negative charge of the phosphates on the DNA backbone. This allows DNA molecules to be more prone to sticking together, because if DNA molecules, if their backbones still have a negative charge, then the same charges repel each other. However, when salt is added, which neutralizes the backbone, then which neutralizes the negative charge, then the DNA molecules can stick together, which then later allows the DNA to precipitate and to become visible in the solution.
Okay.
It's like fishing in a pond.